Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Welcome to the Neurosciences 2 module. This is Dr. Asma Shabir, Assistant Professor from the Department of Pathology, JSMU. The topic of this lecture is the reactions of neurons and glial cells to injury, cerebral hypoxia, and cerebral edema. Learning objectives of this lecture are to explain the types of glial cells, their normal functions, and their reactions to injury, discuss the role of microglia in CNS inflammation and repair, and to describe the pathophysiology of hypoxia and cerebral edema. So there are two types of CNS cells, that are the neurons and the glial cells. Neurons are meant for the conduction of nerve impulses, and if destroyed they cannot be replaced, whereas the glial cells are to support, nourish, and protect the neurons. These include astrocytes, oligodendrocytes, ependymal cells, and the microglia. This shows the structure of a typical neuron representing the cell body and the axon. The cell body has the neurons and the extended dendrites, whereas the long axon is covered by the myelin sheath and the Schwann cells. This slide represents the features of different neuroglial cells present in the central nervous system, the ependymal cells with flat cells and the nucleus, whereas astrocytes with the extending processes, the oligodendrocytes and the microglia, whereas the peripheral nervous system consists of the satellite cells and the Schwann cells. This shows the functions of different glial cells. When you talk about the glial cells in the CNS, number one are the oligodendrocytes, which help in the myelination of the CNS axons and provide structural framework, whereas astrocytes maintain the blood-brain barrier. They provide structural support. They help in the regulation of iron and nutrient concentrations. They even absorb and recycle the neurotransmitters and also help in scar tissue after injury. Whereas microglial cells are the macrophages of CNS, which remove the cell debris by pathogen phagocytosis. Ependymal cells line the ventricles and the central canal, which assist in producing and circulating, monitoring the CSF. The neuroglial cells in the peripheral nervous system are the satellite cells, and the Schwann cells. Satellite cells surround the neuron cell bodies in the ganglia, which regulate oxygen, carbon dioxide, nutrients, and the neurotransmitter levels around neurons in the ganglia. Whereas the Schwann cells, they surround the axons in PNS and help in formation of myelin, and they also participate in the repair process after injury. So the oligodendrocytes help form the myelin in the CNS, whereas the Schwann cells form myelin in the peripheral nervous system. So this shows the hematoxylin and eosin stain for the neuronal tissue, which shows the pointing arrows with neurons and the glial cells. Along with this, you can see the neuropil, an entity labeled as neuropils, which are the mostly unmyelinated axons, dendrites, and the glial cell processes, which forms dense synaptic regions. These are the dense synaptic regions with these processes, and they have a relatively low number of cell bodies. A better view presenting with the neurons and the glial cells, and within the cell body of the neurons you can see the basophilic granules. These are the nasal granules or the nasal substances, which are the rough endoplasmic reticulum. Again, you can appreciate the size of the astrocytes and the oligodendrocytes in this figure. Now, starting with the reactions of neurons to injury, the etiology may be due to acute or chronic causes. Acute, most common, is due to the depletion of oxygen or glucose, trauma, and the chronic cause are the degenerative disorders and abnormal protein aggregates. After injury, these neurons represent with different morphological features. The one with the acute neuronal injury are termed as red neurons, which are evident after 12 to 24 hours after an irreversible hypoxic or ischemic insult. And the histological features are those of the necrotic changes seen in the neurons, with shrinkage of cell body, pycnosis of the nucleus, disappearance of the nucleolus, and there is loss of nasal substance. This loss of nasal substance, the basophilic granules, gives intense eosinophilia of the cytoplasm within the cell body of the neurons. So you can appreciate here in this picture representing the red neurons. These are the red neurons with loss of nasal substance, pycnotic nucleus. Rest of them are the glial cells, these blue dots, and the base is all the synaptic regions representing the neuropil. Here somehow you can see the normal neurons representing here. These are the normal structures where these are the red neurons, and these are the oligodendrocytes, different glial cells, and these are the neuropils, the synaptic processes. Second morphological features after the subacute or chronic neuronal injury is the degeneration. These are the causes include neurodegenerative diseases such as amyotrophic lateral sclerosis and Alzheimer's disease. Histological features are due to apoptosis, whereas there is cell death and there is cell loss. You cannot appreciate any of the neuron in this figure. Instead, you have gliosis. It means that there is proliferation or hypertrophy of glial cells. You can see the arrows pointing here. These are the astrocytes which have proliferated, undergone hypertrophy and hyperplasia. All these blue colored are the glial cells. There is an increase in the number and size of the glial cells termed as gliosis, and there is loss of neurons due to apoptosis in this degeneration. And further, in extreme form, this process of gliosis can lead to glial scar formation as well. Third morphological feature where they can represent as axonal reaction, which are seen the changes seen in the cell body with regeneration of axon. These are showing increased protein synthesis. These are the reactive changes which represent the histological features as spheroids, which is rounding of the cell body. There is peripheral displacement of the nucleus, enlargement of the nucleolus, and most important is the central chromatolysis, which means there is dispersion of nasal granules from center to the periphery. So the axonal reaction here, number one, the figure A represents the red neurons where the cells, the neurons are shrunken due to hypoxia in cerebral cortex. Figure B represents the axonal spheroids. These are the axonal spheroids. These are the different glial cells representing here along with the neuropils. And the figure C represents the chromatolysis where there is the cell body is swollen, pink cytoplasm, the nasal granules are displaced towards the periphery, termed as chromatolysis, a feature of axonal reaction. A better view of chromatolysis, these are the chromatolytic neurons representing here with displaced nasal granules with pink cytoplasm, a clear feature. Now, neuronal damage may be associated with a wide range of subcellular alteration in neuronal organelles and cytoskeleton. The neuronal inclusions may occur as manifestation due to aging, viral infection, or degeneration. Basically, these are the proteins which are accumulated within the nucleus or cytoplasm. In aging, you can see the intracytoplasmic accumulation of complex lipids, proteins, or carbohydrates. In viral infection due to herpes, we see intranuclear inclusions termed as Cowdery bodies. In rabies, the intracytoplasmic inclusion bodies are termed as Negri bodies, whereas nuclear and cytoplasmic inclusions are seen in cytomegalovirus infection. In degenerative diseases like Alzheimer's, we find neurofibrillary tangles. Lewy bodies are pathognomic of Parkinson's disease, whereas you can see abnormal vacuoles deposited in the neuropils in Creutzfeldt-Jakob disease. So this picture, this arrow points towards the cowrie bodies seen in the herpes infection. You can see here, these are the intranuclear bodies composed of nucleic acid and proteins seen in the cells infected with herpes. The negri bodies, the arrows pointing towards the sharp outlined pathognomic inclusion bodies seen in virus infection cells with rabies, and they consist of ribonuclear proteins produced by the virus. Here we can see the feature of Alzheimer's disease, where you have the intra-axonal and interaxonal deposits of proteins inside the cell body. You can appreciate these are the neurofibrillary tangles, which are the aggregates of hyperphosphorylated proteins, which are insoluble twisted fibers and are primary markers of Alzheimer's disease. And these are the amyloid plaques in between the neurons. So within the neuronal cell body and between the neurons, you can see the deposits of proteins in Alzheimer's disease. Lewy bodies, again, these are the aggregate of proteins developed inside the nerve cells in Parkinson's disease. We can appreciate intranuclear as well as intracytoplasmic inclusions in cytomegalovirus infections. So coming up with the reactions of astrocytes, we have discussed the reactions of neurons to injury. Now we talk about the reactions of astrocytes, the glial cells to injury. This represents the picture from Robbins, the astrocytes and their processes and the IHC staining for glial fibrillary acid proteins and the well-developed processes of astrocytes. The astrocytes react to injury in giving different morphological features. The most important, the common one is the gliosis, as I have discussed. This is the hypertrophy and hyperplasia of astrocytes. The etiology of injury might be anything, but it represents with hypertrophy and hyperplasia of astrocytes termed as gliosis. Second morphological feature where astrocytes can present are the demistocytic astrocytes, which you can see in this figure, the vesicular eccentric nuclei with prominent nucleoli and the bright pink irregular cytoplasm. So these are the demistocytic astrocytes which are seen after the injury. Now next morphological feature of astrocyte, they can present as Alzheimer type 2 astrocytes. To remember, it is not related to Alzheimer disease, but instead the etiology lies on with the chronic liver diseases representing with the long-standing hyperammonemia, where the astrocytes show large nucleus, pale staining central chromatin, intranuclear glycogen droplet, prominent nuclear membrane, and nucleolus. Whereas the next important morphological feature are the Rosenthal fibers, which are the common findings seen in long-standing gliosis, whatever the etiology. These Rosenthal fibers are also seen in pilocytic astrocytoma and Alexander disease. What are these? Are the thick, elongated, brightly eosinophilic, irregular structures within the astrocytic processes. So these are the astrocytic processes which have developed pink, irregular eosinophilic structures termed as Rosenthal fibers. So again, a closer view of these Rosenthal fibers, pink eosinophilic structures within the astrocytic process. The astrocytes can also present with certain degenerative changes due to aging termed as corpora amylacia. These are the glycosamine polymers containing heat shock proteins and ubiquitin. What is ubiquitin? These are the small regulatory proteins which helps to regulate the process of other proteins through ubiquitin proteosin pathway. I'm not going in detail because we have covered this in your general pathology in the chapter of cell injury related to ubiquitin. So these corpora amelitia are the degenerative changes within the astrocytes due to aging, which are the round, faintly basophilic, PAS positive, and concentrically lamellated structures located wherever there are astrocytic end process. Elaphora bodies are seen in cytoplasm of neurons in myoclonic epilepsy. Now, next we move towards the reaction of microglia to injury. We have discussed the reactions of neurons, reactions of astrocytes, and the most important here are the reactions of microglial cells. Now, to revise, the microglial cells represent the macrophage monocyte defense system of CNS, which are the small cells of mesenchymal origin. They have elongated nuclei and relatively little cytoplasm, which forms fine, highly branched processes. And in response, now you can see here this is the resting microglial cells. In response to tissue damage, these microglial cells change themselves, transform themselves into large amoeboid phagocytic cells, and the basic function of them are to protect neurons from disease process. So you can see this is the microglial cells which have changed themselves into plump cells and forming the macrophage. They have changed themselves, the microglial cells, into the plump cells which help in phagocytosis. So whatever the infectious agents, neurotoxins, the resting microglial is going to change themselves into activated microglial cells, which in the presence of interleukins and nitric oxides, free radicals, leading to neuronal death. This picture is representing the different phenotypes and the functions of microglial cells. You can see to the extreme here, right, you have the resting microglial cells. Now, after injury, how do they respond? They differentiate and migrate. They differentiate into M1 phenotype and M2 phenotype microglial cells. M1 are the pro-inflammatory microglial cells which change themselves into plump cells, and in the presence of different cytokines and the interleukins, they lead to neuronal damage. Whereas M2 phenotype microglial cells are anti-inflammatory, where in the presence of interleukin 4, 13 growth factors, they help in the repair of neurons. So the different ways they react, the M1 and the M2 microglial cells. Whereas I have discussed about the astrocytes as well. After the injury, they change themselves, they become the reactive astrocytes, and astrocytic gliosis can lead to glial scar formation. Now here to just have certain thought, like certain research questions arise where one can think of novel treatment strategies to protect the neurons. How you can think of certain research questions. How can one think to protect the neurons? Is there any arising novel researches? So I have come across one of the review article which I would like to share, and this has offered the role of mesenchymal stem cells therapy in targeting the microglial activation. You can see this mesenchymal stem cells which releases the paracrine factors. I'm not going detail in what do you mean by paracrine, because this as well you have studied in your general pathology, the autocrine, paracrine, and the endocrine factors. So you can refer those chapters if you need. So they release the paracrine factors which modulate the microglial activation. So what can these do? These can inhibit the activity of M1 phenotype microglial cells. As we have seen in our last slide, M1 are the pro-inflammatory. So modulation of microglial cells can inhibit the M1 phenotype, whereas M2, which are the anti-inflammatory. So this can augment the activity of this anti-inflammatory M2 phenotype microglial cells. This together can augment brain repair by inhibiting the release of mediators driving the pro-inflammatory processes and augmenting the release of mediators which help deriving the anti-inflammatory events. So together can augment the brain repair. So here you can see that the mesenchymal stem cells might improve outcomes of brain injury by inhibiting the activity of M1 phenotype microglial cells and augmenting the activity of M2 phenotype cells. Now, how does the microglial cells histologically respond to injury? Four features you can remember. These are the proliferation of microglial cells. They develop elongated nuclei which are termed as rod cells. They form aggregates around small foci of tissue necrosis which are termed as microglial nodules, and they also aggregate around the cell bodies of dying neurons. These are termed as neuronophagia. So proliferation, formation of rod cells, microglial nodules, and the neuronophagia is the histological features representing in the injury by microglial cells. This picture, this arrow presents over here in this figure the rod cells in neurosyphilis. You can see the lymphocytic infiltration around the neurons here, infection with the viral encephalitis, the microscopic pictures. Here you can see the microglial proliferation, formation of a microglial nodule. This represents the neuronophagia where the microglial cells have accumulated around the dying neuron. This is typically the microglial nodules. Accumulation of microglial cells which have become plumped has undergone changes because they have become phagocytic now. Now we move towards the next subtitle, that is hypoxia and cerebral edema. The brain tissue is composed of water and is separated from systemic circulation by a complex series of interfaces, as you know, the function of blood-brain barrier, the choroid plexus, and the arachnoid plexus. So these have specialized proteins, you can say, that forms tight junctions and prevents the solutes in the circulating blood from non-selectively crossing into the extracellular fluid of CNS. So this is the role of blood-brain barriers which protects the brain. Cerebral edema is the result of increased fluid leakage from blood vessels or injury to various cells of the CNS. It is the end result of many neurological diseases which represent serious life-threatening consequences of many common brain disorders like stroke, trauma, tumors, and infection. Cerebral edema is classified basically, or you can say there are the two pathways of formation of cerebral edema, that is cytotoxic edema or cellular swelling, and vasogenic edema or vascular leakage. The third one is the interstitial edema or hydrocephalic edema, which occurs especially around lateral ventricles, and this is due to increase in intravascular pressure. The basic etiology of cytotoxic edema is due to ischemia, trauma, toxins, and the metabolic diseases, whereas vasogenic edema is most commonly in infections and inflammatory conditions, the brain tumors, and these together leading to hyperosmolar states. Interstitial edema is related with hydrocephalus. Cytotoxic edema is increased in, remember, the intracellular fluid secondary to neuronal, glial, or endothelial cell membrane injury and is induced by stroke, trauma, or toxins. It is represents in 24 to 72 hours after stroke, where there is rapid increase in fluid due to energy failure and there is loss of ATP as a result of hypoxia and ischemic insult. Whereas vasogenic edema is increase in extracellular fluid, which is caused by disruption of blood-brain barrier and increased vascular permeability. Vasogenic edema may be localized around the inflammation or neoplasm, or it might be generalized after ischemic injury. So, this represents the pathophysiology of both the types of cerebral edema. Whatever the cause, that after tissue damage, you can see there is disruption of cell membrane pumps, which leads to cell membrane dysregulation. There is influx of water into the cell from interstitium, leading to cellular swelling termed as cytotoxic edema. Whereas if there is disruption of blood-brain barrier by damage or endothelial cells, there is increased blood-brain permeability, which leads to protein leakage. Along with that, after injury, you have abnormal cell metabolism where there is the cells produce osmotically active products. So abnormal osmotic balance is responsible for influx of water into tissues from capillaries. Remember, extracellular. So there is tissue swelling leading to vasogenic edema. This is the gross morphology of brain representing cerebral edema, where you have the narrow sulci and flat gyri. This last slide represents the pathophysiology of cerebral hypoxia. Just to inform you that hypoxia has been covered in detail in neurosciences one module along with cerebrovascular diseases. But to have an overlook with the pathophysiology, I'm explaining you here. Whatever the cause, the stroke, trauma, inflammation, with ischemia here, what will happen? There is depletion of energy, depletion of ATP, and loss of membrane potential. Which in then, what will happen? In then will activate a cascade of enzymes. Now these enzymes that contribute to cellular injury. There is increase in calcium influx. There is release of excitatory neurotransmitters such as glutamate, and this contributes to the cell damage. Further, what happens that there are cytokines which continue the damage and results in the opening of blood-brain barriers, whereas chemokines attract WBCs to the injury site where there is release of free radicals and proteases, and this enhances the injury. Finally, in the later stages, the proteases, the free radicals, they attack the structural components and together leading to membrane damage and cell death. So wrapping up the lecture with the key concept, reactions of cells to injury, neurons become red and then degenerate, astrocytes undergo gliosis, the microglia proliferates, and the rest of the glial cells like oligodendrocytes and the ependymal cells, they don't react much to the injury. Cerebral edema is the accumulation of excess fluid within the brain parenchyma. It forms by two major pathways, that is cytotoxic edema and the vasogenic edema. So we'll just have in the end certain quiz questions. Let's see if you can solve. Name the neuroglial cells of CNS and PNS. What are red neurons? What is chromatolysis? Name the inclusion bodies in herpes and rabies infection. Describe the features of dermatocytic astrocytes. In what histological ways does microglia respond to injury? Name the two major types of cerebral edema. This figure says that identify and name the pathological change seen in astrocyte processes. You need to identify the pink fibers of astrocytic processes and name them. Thank you and have a good day. Allah Hafiz.